out air out from the other direction, because when the air travels with hot air, so there is a gradient atmosphere that is created, and every 3 meters there is a monitoring system, that is temperature, CO2, RH. So it is all data is tracked and controlled. So we are basically controlling the air velocity and monitoring the whole system. Temperature gradient tunnel we have developed in many places like Ikrisat, CRRI Kotok, IIVR Varanasi, IRI New Delhi. There are so many publications. You can see by using our system, they have developed so many varieties. The fourth technology, we are also making big controlled greenhouse. Greenhouse, you know that it's a big control with temperature controlling option. We have done it for various research institute, including IIPR, Indian Institute of Pulses Research. Recently, we have done a big project at BRC. It's basically a greenhouse. It's a control chamber. It has got 12 cubicles. Each cubicles you can control for temperature, light, and it is controlled through a big PLC. So it is a basically 12 plant growth chamber. You can have different treatments with sprinkler, micro irrigation systems. We have provided the entire system for BRC. The fifth, what we are doing it, we have developed also aerophonic systems, hydroponic systems for different research institute. Like we have done it for Kharagpur IIT. Aerophonic system we have done it for Kerala Forest Research Institute. Fifth or sixth product that what we are doing it, that is walk-in plant growth chamber. We are making big working chamber for many research institute. We have done it for BRC, Bhabha Atomic Research Center. We have done it for Indian Institute of Soil Science. It's ICR Research Laboratory in Bhopal. This is basically for controlling the chamber. You can control temperature, control CO2 and humidity level, and you can grow plants inside the chamber. We have developed many plant growth chamber for TNU, that is Tamil Nadu Agriculture University, and those plant growth chambers are using for maybe tissue culture, some other applications they are growing. So last 14 years we have sold our technologies. These are all indigenous technology. The important part is these are our own indigenous technology. We give the system, we provide, we develop. This is our own designing. Everything that is been taken care, and all systems are supported by us, and we are always trying to do something new. You are into now entering into few new technologies, like developing few technology that is related to bioreactors. Basically, our interest is to develop some good quality bioreactors, and we have already done in successfully have done some bioreactor project for Assam Agriculture University. I believe altogether 6 or 8 bioreactors we have developed for to study azula rice interaction studies. And azula rice, anabina, all who are the microbiologist or agricultural scientist must be knowing, it is helping directly or indirectly for enriching the soil nitrogen level, as anabina with association with azola, the fixed nitrogen, and that nitrogen is helping in enhancing the nitrogen level in soil, which is again directly taken by plants. So in nature there is always a chance to utilize a lot of symbiotic organism and use them as kind of a fertilizer. So we are trying to do with some research institute associated with DST project. Also, we have submitted some project proposal. So we are developing some photobioreactors. So we are trying to minimize the chemical fertilizer. See, if you again go back after 1947, after independence, India government has not given much more emphasis towards biofertilizer. Whatever emphasis that is given for development of chemical fertilizer. Again, if we want to have a natural, the organic way of doing cultures, we can use BGA, that is blue green algae and other algae, and multiply them, sell as a fertilizer. Now you can develop so many strain which are first growing. So there are technologies available. You can have a lot of strain that can be collected from the local sources, multiplied and give it to farmers back. There are many technologies that is available also related to VA mycorrhiza, vesicular or vesicular mycorrhiza, development of culture and developed as a biofertilizer, package it and sell it to farmer. There are n number of entrepreneurship can be developed. A few years back, I was very much associated with Government of India project with ICR for entrepreneurship development skill. I was trying to coordinate with so many scientists, understand their technology, and finally we are making interaction with scientists and farmer. We are making techno-economic business feasibility report, and we have submitted many proposals to Government of India as well as to ICR through ICR. So those things are helping us now. Those technology, being an independent entrepreneur, we are trying to serve the similar way. Now coming back after many years, if you really look back and analyze what what kind of impact probably it is generating as an individual, as a company, when you run a company, you have like know that impact analysis. If you want to study it, that impact, it is very difficult to quantify it, but nevertheless the kind of systems we have provided, almost 60 projects. Now if you imagine each project is associated with minimum 3 or 4 scientists, some research students, a group of 10 people. It's basically a kind of an association of close to 600 scientists, includes the research student, maybe JRA, VSRA, RA, some postdoc students, they are involved. There are so many publication. It's basically those kind of system is helping to understand how the different field people can work together, because any biological system or any simulation modeling system when you work on, there is every possibility. There is a need for a biotechnologist, there is a need for a microbiologist, there is a need for a plant physiologist, there is a need for a taxonomist also. This is the beauty of doing interdisciplinary science, where you can have much more closer interaction, and this is required for the betterment of science. Again, I strongly believe a total science can be achieved, its goal towards the benefit of common man, towards the benefit of humanity, or towards the benefit of the living world, is to work together, and always think beyond certain things. Whether if you want to bring something in place, you have to have the combination of all those ideas, and it can be put into one platform. Sometimes this idea might be wrong, some ideas might not be accepted, some would be rejected. That is the part of science. So science is nothing like that whatever you said, but whatever I said, it is the final thing. After a few years down the line, you will see the same thing. What we used to think is right, maybe it has got different interpretation, and it has got no relevance. It is very much similar to writing a poetry or a novel. You write today something, you keep it. After a few years, you take out, you see that some words are very much irrelevant. You don't need it. So it's basically time and the perception what you have at that particular time about you have written. Now, after many years, you might feel those are not the relevant thing that is what is required. This is the kind of part of evolution. That's why if you see that the great literature is today is surviving. If you take Shakespeare, Shelley, Keats, Byron, Rabindranath Tagore, or Jivananda Das, any sense, or Guljar, Ghalib, everybody, like you see that they have not written much, but whatever is written, it has got its own impact till after so many years. That's why we calculate the entropy values. Like so many people are writing English literature till Shakespeare's literature holds so much importance in our life. Why? It is because of the choice of words. Each word, like no, if you take his just write up, you cannot eliminate a single word. If you take out one word, the importance of this particular content will be much lower. Similar way, our research and development, like no, each and everything, our activities, you can literally judge it many ways. So friends, it is needless to say that what we have done or what I have done, because I always believe one thing, that is is all the world is a stage, and all men and women mere players, they have their exits and entrance. So everybody has to go one day, but I personally believe that if you can leave something for the next generation to do it, to think beyond. So you don't do the traditional science, I always believe. Try think out of the way, think something funny things, like and that is going to give you more opportunities to think. If you think traditional way, you might stuck up somewhere. So I'll end up saying that our this coronavirus days also will be going out. Will scientists, everybody will try to do something to defend this coronavirus, develop some vaccine, develop some immunity kit kind of thing to counter them. And contrary, my opinion is very simple. This is my again suggestion, my thought. This coronavirus is not a kind of a spontaneous origin of this species. It is there very much. It is modified. Either it is a new mutant, or somebody, like, no, I really do not know, somebody has developed a new mutant and which has been multiplied and spread. Somebody might have the political interest. This world is a very complex. There are so many countries, people are having nuclear weapon. Now people are saturated with nuclear weapon, right? There are many countries. You think of the big giants: America, Russia, China, Israel. So all those countries, France, they have all kind of nuclear weapon. North Korea. Now each one is feeling that they're saturated. Now, if somebody, it might happen that one day, if somebody has got very ill intention to destroy the whole world, it can be done within 5 minutes. Things are very tricky in a political and economical situation where the global population is increasing, aggression from one country to another countries. Everybody is looking one to another in a different mode. The relationship is not human. We are not thinking as a total human species to save the global world. Again, I've been talking from the beginning that we should have rational thinking and a holistic approach to do the science. The science can be used for destruction of total human world. The real example is Second World War. The purpose of developing nuclear, uranium, and other things, and you know the history of Otto One. And after that, and when America established Manhattan Project during the Second World War, and they have finished Hiroshima and Nagasaki in 1945. So what are the reactions from the scientific community that I also know, all in public domain? Same way, the biological war. We have been talking, we have been hearing this biological war. It's not very few years back, way back in when the Iraq war takes place, when Saddam Hussein was attacked and eliminated by America, by friend UK. Their statement was there, the anthrax was there. The Iraqis are making anthrax to kill the common people. And this is the only way to attack Saddam Hussein in Iraq. And what is the outcome? The outcome is obvious. There are lakhs of Iraqis have been just stoned to death. The entire country has been destroyed. But they have not found the anthrax. So some biological tool also are being used to destroy the countries. I am nobody, I'm not an expert to talk about all those issues. But if there is any war, there is possibility of any kind of war, it is the biological war, and it is a very silent. It is very difficult to tell that you have done this job. It is not that easy, because as I have told you, microorganism is a special creation by God. It is invisible, it is microscopic, even you cannot see in normal microscope. You need to have electron microscope to understand. The virus typically in nature is a dead particle. When it is not contacted with the host, it works as a dead body. Is like only is its physiological or biochemical activity starts, its multiplication starts when it is in contact with the host. That is also the specific host. So one virus may not be relevant to another host or living organism. So for them it is kind of a dead particulates. So friends, what I'm trying to tell you here is the scientific community, or if somebody is forced to do some kind of research which is not for the people and for the vested interest, it will be more dangerous in future days. And it is very difficult to say the human world as a whole. We are all threatened, and there is a kind of a danger signal that is going on. There will be always suspicions. You have done it, you have done it, you have done it. But ultimate thing is we need to save the humanity, the human world together. So I wish I have not told anything wrong. This is my personal feeling. You can have your own opinion, but you always good to speak. But if you do not speak, then who is going to speak? So again, I wish we shall overcome these days, and we have overcome